Thank you. I just wanted to give you some examples of our work that hopefully informs a little bit the debate about using omics for understanding the function of human genetic variants. And really, over the last 10 years, my work has mostly worked not on disease, but in healthy population cohorts, and really trying to understand how we can use different layers of information to annotate the function of genetic variants. And I just want to give you 3 examples of some of the work and where this is leading us. So the first is really actually taking a standard GWAS framework to learn some of the rules of complex trait associations. We work a lot on blood counts and blood traits as really a paradigm example of complex traits more generally. Obviously, variation in the cells and the properties of blood cells are important for a number of human diseases. It's obviously a very well studied developmental model. But the other advantage is that the genetics can be easily characterized because phenotypes are really defined at the level of individual cells. And also, it's quite easy to work with individual cell types in physiologically relevant contexts. So we've done over the course of the year many genome-wide association studies. This is all unpublished data, and this is the latest that we've done on the full UK biobank, as well as transethnic samples with collaboration from the BCX consortium. So this is a really highly powered data set. We looked at 29 different phenotypes of blood cell formation and function in about 800,000 individuals. And we were able to identify a really large number of associations. There are over 10,000 independently associated variants that we discovered in this data set, including 500 rare variants below 1%, and indeed many below 1 in 10,000 in the population, and 820 that we call low-frequency variants. And we begin to really understand and study the properties of rare variant associations. So first of all, 95% of variants overall are non-coding, but indeed, remarkably, also 65% or so of the variants that are rare map to non-coding elements. And many of these variants have huge effect on phenotype, greater than 0.5 standard deviation, but they are indeed non-coding variants with effect sizes of over 1 standard deviation on the population mean of the trait. So being very well characterized data set, we can do fine mapping actually quite easily. So about 10%, so 1,000 associations are fine mapped to a single credible set of a single causal variant. And indeed, nearly 50% of the associations, so about 5,000 associations, are mapped to less than 10 causal variants. And what is remarkable is that compared to many other phenotypes, we are beginning to really have a much greater predictive power by only taking into consideration the sentinel variants or the fine mapping sets compared to genome-wide variants. And this really suggests that we're at the point where we probably have captured the majority of predisposing loci in the population. So as I said, being a well-established system, we can begin to look at the function of these variants in cell-relevant context, and this is really beautiful work by Vijay. And so what he did is mapped a single-cell resolution, the chromatin accessibility using ATAC-seq for all the cells that are relevant to our phenotypes, as well as all the progenitor steps. And so what you see, for instance, if you take a set of variants associated with red blood cell count, about a quarter of them map to open chromatin regions that are only open in the terminal differentiated state of the erythroblast, of the erythrocytes. Many map to variants that demarcate progressively enhancers that progressively open along the developmental trajectory. But indeed, many others are in regions of open chromatin that are ubiquitously open in hematopoietic progenitor. And there are many other cases, indeed, where the enhancer is open, but then it closes up later on. And so we can really begin to fine map, to take our fine map variants and map them to their individual enhancers. And this is just one of the examples. A variant that maps to an enhancer in BCL2 gene is associated with low decrease of red blood cell counts as well as monocyte counts. And we see very concordant patterns of chromatin state in the relevant cell types. So this is a phenotype where we can quite easily go in the lab, at least relatively easily go in the lab, and find map mechanism. And Vijay has developed an impressive array of techniques that allow you to interrogate the function of variants and genes. And this is just an example of two independent variants. About 20% of the signals have independent variants that map to a region of open chromatin. And as you see, the gene expression phenotype, the enhancer activity recapitulates quite beautifully the original phenotype for which we discovered these variants. So one of the really the point that we're always thinking about is how does this fit in the context of a broad understanding of human pathways. And George has already alluded to the omnigenic model. Really here the idea is that there should be a set of core genes or genes that are potentially disease-causing and a set of interacting genes. So what we did is to try and begin to see whether our extensive GWAS data fits this model. And so what we did is we annotated manually curated all genes implicated with Mendelian forms of blood disorders, including platelet disorders, red cell disorders, and myeloid disease. And then we intersected this data with a very conservative estimate of the annotation of our fine-mapped variants to genes. And what you see is that, first of all, and this has already been shown many times, is that GWAS variants are significantly enriched within Mendelian disorders. But what was quite surprising to see is that the genetic variants that are not reported disease alleles, but are within these genes, tend to have much greater effect sizes and contribute disproportionately to the predictive power of the genetic score. And this is not only true for missense variant, but also for regulatory variants. So there is a possibility that indeed these genes might harbor variants of clinical interest. If you take indeed the other genes, so the ones that do not have a Mendelian disorder, and you annotate the connectivity from blood co-expression network, what you see is that these genes are significantly more likely to be interacting in a co-expression network compared to the Mendelian genes. And so this really bears the possibility that there are two classes of genes, or indeed classes of variants, one that performs a homeostatic function, if you want, in regulating blood cells in counts in health, and the other one that indeed causes variation. And one of the corollary to that is that it is possible that hidden between our association, there are variants that are not directly disease-causing, but indeed modify disease phenotypes. And this actually, the principle has been shown quite nicely very recently by Peter Campbell and colleagues, who showed that they sequenced indeed array genotyped a series of cases of patients that have two myeloproliferative disorders called essential thrombocythemia and polycythemia vera, which are characterized by an excess of platelet on this axis or an excess of red cells on this axis. So the causative mutation for these diseases is JAK2, but what we're able to show that some of the variants that we had previously identified, the reduced hemoglobin or increased platelet count, are indeed modifying the cancer phenotype towards a higher frequency of ET. And this clearly also informs treatment options for these patients. So there are many, I think, lessons learned from blood cells, and what I certainly want to emphasize is that even understanding of the exact cellular context that is relevant for phenotype is not sufficient, is not enough to really use a regulatory annotation to annotate disease variant. But there's certainly a possibility of indeed exploiting this sharing of regulatory annotation to enhance interpretation. So the second, third vignette are much shorter. So first of all, we wanted to see, okay, if there is sharing of molecular annotation, can we use this information to improve our ability to find map disease variants? And so we know very well that many, for in this case, autoimmune diseases share very much, are both comorbid, but also share underlying pathophysiological mechanism. So when you want to find map from a disease GWAS, I think that you're faced with the fact that disease GWASs are inherently very noisy. So they have, individual variants have lower odds ratio. They are typically meta-analysis of multiple study with a lot of underlying heterogeneity in the way the samples are processed. There's heterogeneity in summary statistics. It's hard to match LD from summary statistics, and also you're dealing with one phenotype at a time. So over the course of time, it's been a realization that indeed molecular QTLs can be overlaid to disease associations to really try and gain some confidence that there is a sharing of genetic effects between the molecular phenotype and the disease phenotype. And dealing with molecular phenotypes is much easier because, of course, these traits have higher effect sizes. They're typically much more homogeneous in the way they've been generated. We have complete summary statistics, and indeed, you can match very well linkage disequilibrium to the summary statistics to deal with regions with complex associations. So we exploited this possibility to ask a simple question, which is, if you have a multi-omic QTL data set, can you find map, can you co-localize this with the disease variant? Can you find map the causative variant better than if you were, you could do if you were working with a summary statistic alone? So what we did here is a very large scale co-localization effort where we took summary statistics from 14 different autoimmune diseases and 5 molecular phenotypes across 3 immune cell types. We co-localized systematically with high posterior probability and identified 346 loci where we have a co-localizing molecular and disease signature. And then we said, what does the fine mapping on the molecular QTL tells us that the disease doesn't? And so this is an example of a variant in integrin 4. If you find map from the disease summary statistics, you have a credible set of 11 variants, and the top variant that is deemed to be the more likely to be causal is a variant down here with really no information on its function and a low posterior probability. So if you add histomodification data, so we find map on the histomodification, you see that this quickly reduces the credible set to only 3 variants. And indeed, the gene expression data converges on 2 variants as the more likely causative ones. And indeed, one of them disrupts the binding for CBP beta, which is a relevant transcription factor binding site. And this is really an exemplar picture, but we see this happening over and over again. Molecular QTL, given a similar density of association, find map a lot better than disease association. And indeed, in 50% of the cases, molecular QTL show that the likely causal variant is an indel that was completely missed in the disease associations. Do you have a minute? One. Okay, the third very quick example is thinking back about the layers of molecular or data that we're generating. Obviously, when we work in large cohorts, we're limited by phenotypes that have a sufficiently high throughput or low cost. And the question is whether there are opportunities to develop phenotypes that are perhaps more relevant to the ultimate function of the cells. And in the case of blood cells, very relevant phenotypes are the granularity of cells, so the amount of proteases that influence immune function. And so with colleagues, in particular with Willem Hoverand, we have looked at ways to re-hack, if you want, Sysmex automated analyzers to derive new measurements of cell properties, including cell size information, cell structure, which indeed reflects cell granularity and the permeability of the cells. And so these are very cheap to obtain. They are just simply a Sysmex full automated analyzers, but we think are much more relevant to cell function. And indeed, if you do association studies and then you overlay this information with disease variants and genes, you see that there is significant co-localization. And this suggests that we're able to capture molecular, cellular functional phenotype in high throughput manner. So just to conclude, just to say we, at least in our phenotypes, we're really at the stage where we have a near complete catalog of genetic variation and their effect in humans. We have a good idea of comprehensive characterization of the allelic architecture of these traits. And while it's relatively easy to do high throughput functional screen, there's a number of questions that are indeed continue to be relevant for many complex traits, including how we deal with how we deal with, for instance, data sparsity, collecting large homogeneous sets of functional data for integration, and how we plan downstream experiments from this. And with this, I'd like to conclude many collaborators that worked on this project, and John is here in the room. Thank you.
Thank you so much. A wonderful tour de force. That's really big data right there. Any questions for Nicole? Eric?
Great talk. Thank you. Thanks for all the overarching work over the years. You know, passing reference to the core gene notion. And I was trying to wrap my head around, is it like a well-defined notion? I know it's statistically defined as if everything else was held constant and you change this, it wouldn't matter. And I'm curious between you and maybe George Davies Smith, how do we ever know, is this testable? Is it testable from observational data, where we can't hold everything else constant in human cells, but maybe in your setting we can with isogenic editing. So if this actually means something, then it would mean that if we isogenically delete a copy of the gene or 2 copies of the gene, it will have no effect on the phenotype. That would be a real test, and maybe your hematological traits experiment is the place to find out if there are core genes, or if this is a meaningful definition at all. Yeah. Do you think you have maybe the best place to test this notion in that way?
So I'm very glad you're saying that, because this was my agenda for coming to this meeting. I think this is a perfect model to test some of the assumption that maybe disease settings are very underpowered to test. So there are many. So in this way, we really didn't set off to test the omnigenic, but actually we're reverse engineering. We're saying these are the disease-causing genes, and there are clear differences. So it'd be interesting to see whether we can discover these genes from a network not knowing what they are, right? And so this would be the first thing to do. But so if we take these genes, they're clearly less connected to, there's clearly evidence for this hierarchical order.
Oh, wait, now press back on that they're clearly less connected. There's a difference between the alleles we observe in a population and what the connections actually are.
Yeah. So this is taking just simply the number of edges in the node. So this is a gene level information. Yeah. So I think with VJ, we're really hoping to be able to do systematic screens so that actually some of these hypotheses can be validated in a well-controlled model. Yes.
Cool.